Good afternoon, everybody. Thanks for coming to another Wednesday seminar with the Center for Blood Research. I'm happy to introduce someone who I met just minutes ago for the first time, Mike Gordon, who's a professor in zoology. He did his postdoc at UC Berkeley and his PhD at Stanford and his undergrad at McMaster University. He's and his laboratory are going to tell us about flies as a model system today to learn about biochemistry and sensory perception. And I'll leave it up to Mike to tell us more about the individuals in his laboratory. So thanks very much for joining us today, Mike.
Okay, thanks for the introduction. And yes, as promised, there will be other members of the lab coming through and giving little talks today. I'm going to lead things off, and then we'll each introduce the next person as we go through. So as Ed mentioned, we use Drosophila, and we're interested in taste. And so the overall title for today is Taste Detection and Processing in Drosophila, but I'm going to start out with just one specific little vignette about salt taste. Okay, so one of the nice things about studying taste is that conceptually it's actually pretty simple. So there's tastes that we like, as all animals do, and those tend to be sweet and umami. And the reason why we like these is that they serve as really reliable indicators of specific nutrients that we need, okay? So sweet taste is a reliable indicator of carbohydrates like sugars. Umami taste is reliably an indicator of protein. On the other hand, there are tastes that we don't like. So those include bitter, which can signal the presence of toxins, and sour taste, which is acid taste, which can tell us that food is either underripe or spoiled. But the one real enigma in the taste world is salt. And the reason is that sometimes we like it and sometimes we don't. And really what it depends on is the concentration. So at really low levels of salt, like the salt that you sprinkle on French fries or on potato chips and these other things that we eat all the time, it's delicious. But if you've ever experienced, for example, taking a mouthful of seawater, you understand that high concentrations of salt are really disgusting. And so it's this flip between attractive versus aversive that really makes salt kind of unique among the tastes. And hopefully it's not surprising that Drosophila, our model of choice, behave the same way as we do towards salt. So as you can see at lower concentrations of salt, if we're measuring preference between salt and basically nothing, they will increasingly be attracted to the salt until a point after which there's this dramatic switch to aversion. And the way that salts do this is through the detection of salt through taste. And the way that flies taste is essentially using these taste bristles that are located on different parts of their body. So those include the labellum, which is the tip of the proboscis, the structure that they use to eat. You can think of that kind of like your tongue. But flies also spend a lot of time walking around on the food they eat. And so there's taste neurons also in their legs, as well as places like the wings and even on the female ovipositor. So within each of these taste bristles, generally speaking, there's 4 taste neurons. And we like to think of the information of taste as being sort of categorical. Okay, so one of these taste neurons in a bristle will respond to one category of taste, which we call a modality, right? So there'll be like a sweet neuron, there'll be a bitter neuron, et cetera, et cetera. So those neurons project to a part of the brain called the subesophageal zone up here, which I'm not going to talk about, but you will hear about from others later. So this sort of categorical way of detecting and encoding tastes in fly taste bristles is really analogous to the way it works on our tongue as well, where you can see that in each taste bud, there's a variety of taste receptor cells that respond to different modalities of taste, like sweet, bitter, umami, sour, these things that I introduced just a little bit ago. So in flies, over the years, we've accumulated the knowledge that essentially every taste neuron on the fly can be categorized into one of these 5 groups. Okay, and so these groups are water neurons, and those respond to low osmolarity. So they'll respond to water, but their response decreases as you increase the concentration of any solute. A class that we call iron 94E for a receptor that's expressed in those neurons. We don't know a lot about these neurons, but they seem to respond to some amino acids. There is a sweet neuron that responds to sugar. There are bitter neurons that respond to bitter compounds. And then there's a class of neurons that I'll just call high salt neurons because they're relatively specific to responding to high concentrations of salt. And so for the purposes of salt attraction versus avoidance that I introduced earlier, the way it works is essentially that, that sodium at a relatively low threshold will activate sweet neurons. So that might seem a little weird, but indeed, essentially low concentrations of sodium taste sweet to the fly because they are activating sweet neurons. And that leads to attraction. Whereas as the concentrations of salt go higher, and this is true of all salts, not just sodium, they will start activating these other 2 populations of neurons, which are aversive, the bitter and the high salt neurons. Those drive avoidance, and that avoidance at a certain concentration threshold will override the attraction. And that's why we get this switch from being attracted to the salt to avoiding it. So we can see some of these properties if we look at the activity of neurons in the fly brain. And we do this using calcium imaging typically, where we cut a little hole in the fly's head. We're expressing a calcium indicator called GCAMP, which is a mutated form of GFP that responds to calcium levels by increasing its fluorescence. So we express that in specific taste neuron populations, and then we apply a taste to the labellum and measure the change in fluorescence in the terminals of those neurons in the brain. And so if we look at sweet neurons, for example, you can see that they will respond to increasing concentrations of sodium chloride here. They do not respond to potassium chloride, because remember, these are specific to sodium salts. They respond to sugars, of course, but not to lobeline, which is a classic example of a bitter compound. Bitter neurons, on the other hand, respond to increasing concentrations of sodium chloride, but they start responding at a higher concentration, right? This is one of the defining characteristics of a high salt cell, because you don't want to be activating these aversive cells at lower concentrations of salt. And the other really important sort of hallmark of these high salt cells is that they respond to potassium as well as sodium, and they respond to other salts as well. Of course, they respond to bitter and not sugar. High salt neurons respond much like bitter neurons to these stimuli that we are showing here. Increasing concentrations of sodium chloride, they respond to potassium as well, but they of course do not respond to bitter because these are dedicated high salt neurons. Okay, so as I mentioned, the sweet neurons are what makes salt attractive. So we do a lot of these experiments in the lab where we measure preference, basically by having 2 different choices of little agar dots in a vial. One of them is colored blue, one of them is colored red. You can then let flies eat what they want over a course of a couple of hours, and just then count the number of flies that have a red abdomen versus blue abdomen, and that gives us a measure of preference. So you'll see this in from other people today as well. And so if you give the flies a choice between a low concentration of salt, like 50 millimolar, and water, they are attracted to it, but not so much if you silence sweet neurons. Okay, we do this just by expressing a channel that makes them not fire anymore. On the other hand, if we increase the concentration of salt to 250 millimolar, we can get the flies to avoid it, but that avoidance is reduced if we silence either bitter neurons or high salt neurons. And so that brings us back to this model that I showed earlier, where essentially it's this balance between attractiveness of the low salt concentrations and aversion towards high salt concentrations. So after we learned this a number of years ago, we became interested more in the actual molecules, the receptors that are detecting salt in the environment. And so the rest of what I'm going to show today is the work of a recently graduated PhD student, Sasha McDowell. And so what Sasha did first was to look at the expression of this receptor, a candidate receptor called IR7C. And this is the tip of the proboscis called the labellum, labeled in a fly. And you can see labeled in green are all the high salt neurons. And then labeled in magenta are neurons expressing IR7C. And there's substantial overlap between these populations. So IR7C is expressed in high salt neurons. And we made a mutant to IR7C. And this is calcium imaging, where you would normally see responses to sodium and potassium and magnesium and calcium. And in the IR7C mutants, you see that we've specifically lost the responses to sodium and potassium, but not magnesium and calcium. And so IR7C is specifically required for monovalent salt detection. The difference between monovalent and divalent salts is an interesting question that we are sort of thought about a lot, but I'm not going to talk about it today. So IR7C is a member of this really ancient family of chemoreceptors called the ionotropic receptors, okay? Specific family of receptors that's been given a very generic name, right? Ionotropic receptors can describe tons of receptors, but this is an actual family and we call them the IRs, okay? And the way that these IRs work is that each complex of IRs that responds to a specific stimulus is made up of both co-receptors and at least one tuning receptor. And so co-receptors, which in the taste world are IR25A and IR76B, these are expressed very broadly in taste neurons. So almost every taste neuron expresses both of these receptors. But then the tuning receptor is what actually confers the specific response to that complex, okay? So IR7C is an example of a tuning receptor. And so the model would be that IR7C in combination with IR25A and IR76B creates a receptor that's specific for monovalent salts. Okay, so Sasha did more calcium imaging. There's a lot of bars here, but the take home is actually quite simple that in mutants for either IR76B or IR25A, these two co-receptors, you lose all responses to salt. Okay, so these two co-receptors are required for both monovalent and divalent salt responses, but IR7C is specific to the monovalents. Okay, so there they are. Here's our presumptive complex between IR7C and these other two receptors. And so you would predict then that if we moved IR7C from its normal expression in high salt neurons into sweet neurons, which normally don't express it and normally only respond to sodium, that we would get IR7C-like responses in sweet neurons, right? And this is what we see. So when Sasha did calcium imaging of sweet neurons, normally you can see that they do not respond to potassium salts, but when you express IR7C in them, which are the purple bars, now they do. Okay, so we've conferred this less specific salt response to the sweet neurons by expressing IR7C. And so this has led to this model where IR7C is important in these high salt neurons for detecting monovalent salts. Monovalent salts also activate bitter neurons. We won't worry about that for the purposes of today. But while our Sasha was doing this work, conveniently, another group, the Carlson lab at Yale, discovered a very similar phenomenon happening in sweet neurons. And in this case, the specific receptor was called IR56B. And so that is the tuning receptor for the sodium-specific channel in sweet neurons, which is in contrast to this high salt receptor made from IR7C. And notice that they both utilize these two co-receptors. Okay, so we now have these more of a handle on these pathways. And so one of the things that we became interested in is, the setup of this system where we get attraction to low concentrations of salt and avoidance of high concentrations of salt presumably has evolved so that we eat a reasonable amount of salt, right? You eat things that are low in salt because we need salt, but we avoid things that are high in salt because we can't eat too much or it's unhealthy. But there's got to be more to maintaining homeostasis than that, right? Sure, we excrete salt at different levels and whatever, and that can control how much salt we have in our bodies. But we presume that also there is some fine tuning of the taste system to make us more attracted to salt when we're salt deprived and less attracted to salt when we are salt fed. And that turns out to be true downstream of high salt neurons. In work that we did a number of years ago, we saw that salt deprived flies downregulate essentially the aversiveness of activating the high salt pathway, and that allows them to eat higher concentrations of salt when they need it. But when Sasha was doing some behavior experiments, she saw something interesting, which was that if we compare salt-deprived flies with salt-fed flies, you see this in wild-type flies, we see this big shift in the preference towards 100 millimolar sodium chloride, right? Salt-deprived flies are attracted to it, salt-fed flies avoid it. If you look at the same thing in IR7C mutants, which have lost this high salt pathway, first of all, they're much more attracted to salt because they've lost the high salt pathway, the aversiveness to salt. But secondly, you see that there's still a difference between salt-fed and salt-deprived. And so what that indicates is that in addition to modulating the high salt pathway in response to salt diet, there must be another pathway that changes when the fly eats salt. And so the obvious choice would be to look at the attractive salt pathway in sweet neurons and see whether that changes in response to salt diet. And so it does. This is calcium imaging of sweet neurons now in flies that are either fed on a regular food diet or a food diet supplemented with 50 millimolar sodium chloride. And then if you subsequently, after feeding them on this diet for 3 days, now activate these neurons with different concentrations of salt, you see that the flies that have previously eaten a lot of salt have a much lower response, okay? So they've turned down their sensitivity to salt in response to their higher salt diet, making them less attracted to salt. So we wondered, could this be just sort of a desensitization of these neurons? And Sasha did a number of experiments to look at this. I'm only going to show one of them, which is that instead of feeding them on a high salt diet, which basically chronically exposes these neurons to salt and could cause them to desensitize, she just took flies that were fed on a normal diet and injected salt into their abdomen. Okay, and so when she did that, she saw that salt injection into the abdomen, which is shown here in this these darker blue bars, replicated the phenotype of salt feeding, suggesting that it's really an internal sensor for higher concentrations of salt that then somehow turns down the activity of these salt neurons or these sweet neurons in response to salt. And so we wondered about the specificity for that particular attractive salt taste. And so Sasha looked at flies where we're expressing now IR7C, our high salt receptor, in sweet neurons along with our calcium indicator, to ask the question, okay, in these same neurons that get turned down in terms of their sodium responses, which would be their attractive response to salt, do we see a change in the response to potassium? And the answer is no. So this response is specific to sodium, and so we presume that it's somehow regulating the IR56B receptor. We've looked into a number of mechanisms. I'm not going to talk about them today because there's no time for it. But this is something that we're very interested in as well. And so finally, just to end with the model, we have these sweet neurons that are activated by sodium via R56B. That elicits attraction to salt. Then if a fly has been fed with salt chronically prior to the exposure here, then this turns down the IR56B response via some sort of interoceptive mechanism. And so I'll just end here by again mentioning that Sasha was the one that did most of the work today. And the earliest salt work in my lab was done by two former members, Ali Jager and Molly Stanley. And here are some funding agencies. And we're going to take questions at the end after everybody's spoken. For now, I'm going to introduce the next speaker, which is a PhD student in the lab, Zach Marshall, who's going to talk about actually another taste that is a little bit unusual in that it can be both attractive and aversive, which is sour. So Zach, come on up. Sure. Yeah. Okay. Cool.
Sweet. I'm Zach. I'm going to be telling you a bit about lactic acid taste, a sour tasting substance in insects, and essentially why we should care about sour taste and lactic acid attraction and how it works, basically. So lactic acid is actually a host cue for mosquitoes that spread diseases such as malaria, dengue, and yellow fever. And so mosquitoes, much like fruit flies, are tasting things with their labellum, and they're tasting things with their legs as well. So presumably when a mosquito lands on your arm, it tastes the lactic acid that is found in your sweat. And it uses this basically as a cue to find a food source and make the decision to bite you and take your blood. And so we want to understand how lactic acid taste attraction occurs in mosquitoes, because this could potentially inform, pest control, disease control, and it's also a pretty interesting and exciting mechanism to have the opportunity to learn about. So broadly, I'm really interested in what are the mechanisms for lactic acid attraction in insects, not just mosquitoes, but also Drosophila. So as Mike mentioned, they're a really good system for studying insect taste biology. And this is because they're really genetically tractable. So we have a bunch of different lines that we can use to interrogate how the receptors and the neurons function. We have access to a whole connectome of the brain. So essentially a whole map of every single connection in the brain. So we can start to look at upstream neurons of these peripheral taste neurons. And then also there are a lot of conserved genetic mechanisms between Drosophila and other insects like mosquitoes. For instance, these ionotropic receptors that we are most interested in. So how does lactic acid taste attraction work in Drosophila before we even get to anything related to mosquitoes? So sweet neurons are also required for lactic acid attraction. When we do this two-choice assay where we give the flies a choice between lactic acid and water, and we silence a bunch of different types of neurons, specifically the sweet neurons cause decreased attraction to lactic acid. So it implies that, lactic acid tastes sweet to flies and they're attracted to it. And we think that this is because, lactic acid is a product of fermentation and it's found in fruit. So it signals the presence of beneficial microbes that can ensure the survival of the fly. And so in terms of receptors, we've already talked a little bit about co-receptors. And so IR25A is one of those co-receptors. And lactic acid taste actually involves IR25A. And so when we perform this calcium imaging where we're looking at the projections of these taste neurons and we're applying a stimulus, we can actually look at two different distinct portions of a response when we give a lactic acid stimulus. So there's an onset when the taste is applied to the labellum, and then there's a removal when we remove the taste from the labellum. And we see that the onset response is much higher than this removal response. And specifically in this red box here, when we get rid of IR25A, we actually see a dampening of the onset response, which implies that IR25A is required for lactic acid taste detection. So IR25A is very broad. It's a broad co-receptor. It's involved in a bunch of different tastes. What's the tuning receptor that works with it? So our lab identified IR56D, which is a tuning receptor, and we performed a screen of a bunch of different ionotropic receptors. And so we found, I found that when we knock out IR56D, we actually see that flies avoid lactic acid instead of being preferential to it as compared to our wild types and our mutants. And so essentially, when we have normal functioning IR56D, we see preference, but when we mutate it, we see avoidance. So we want to know how this mechanism works. And once we identified IR56D in flies, our next question is there a functional equivalent in mosquitoes, and how could we possibly identify it? So how we did this is we first performed what's called an AlphaFold analysis of the IR56D protein. And so essentially when we do this, we're just looking at the structure of the protein, because a protein with a similar structure in one species compared to another, they likely have some sort of similar function. And so we identified a bunch of different mosquito ionotropic receptors that were positive hits for the IR56D AlphaFold, and then we looked for receptors that were specifically expressed in the legs. And so here are two proteins. We have our IR56D in melanogaster, and then our Aedes aegypti IR7D, which Kevin, a postdoc in our lab, is doing some work on right now. And so I'm going to talk a little bit about these things. So essentially what Kevin did in these first experiments when he was trying to test some of these candidate ionotropic receptors, what we did was we used the Gal4 UAS system to drive different mosquito receptors into fly taste neurons. And then we were stimulating with lactic acid to see if these neurons gained a response to lactic acid that they didn't have otherwise. And so Kevin identified Aedes aegypti IR7D as being a potential candidate for lactic acid detection. And you can see this through the screen. So we screened a handful of different receptors, and you can see that specifically when we stimulated with lactic acid, that black bar as compared to the green bar, you can see that there's a statistically significant difference in the change in activity in these taste neurons, which implies that this receptor is actually doing something to detect lactic acid and to change the activity of these cells. And so now we want to show a role for IR56D, the melanogaster taste receptor, and the mosquito receptor potentially functioning to taste lactic acid in the sweet taste neurons that lactic acid detection is important in. So we expressed either the melanogaster receptor or the aegypti receptor in sweet taste neurons. And then we were essentially looking in our mutants for the lactic acid receptor to see if we could get a restored lactic acid response. So mutants, we'd expect to see no response, but if we're able to rescue it, we'd expect to see that the these cells have gained a response to lactic acid. And so Kevin performed this calcium imaging. And again, we see that lactic acid responses are higher in our IR56D rescue in the Drosophila receptor and in the mosquito receptor as compared to our mutants at 2 different concentrations, which basically suggests that these receptors can function within the sweet taste neurons that they're important in. And yeah, it really shows that we can also use Drosophila melanogaster as a system for starting to identify some of these receptors. So next, what I wanted to do is we were able to see in calcium imaging that we were able to get a response from both of these receptors. And I wanted to express the fruit fly receptor and the mosquito receptor just within the neurons that IR56D is normally in. So these are the lactic acid sensing neurons. And so what I did is I did a 2-choice behavioral assay, and I gave flies either the melanogaster receptor or this mosquito receptor in the context of the IR56D mutations to see if they would have lactic acid attraction that they wouldn't have otherwise. And so here's my data here. It still is in progress. I'm still collecting it, but we see that my 3 groups in the middle labeled by the Bs, these are my mutant controls. They have a disrupted preference for lactic acid. When we rescue it with the melanogaster receptor in the bluish teal color, we see that we have a restored attraction to lactic acid. And the Aedes aegypti receptor in the green is looking like it could be a partial rescue. It's not statistically significant, but I'm in the middle of collecting this data, and it's possible that this could actually pan out to being a partial rescue, which would be exciting. So we'll see, but the conclusions I have are essentially that IR56D functions as a taste receptor for lactic acid in Drosophila, and that this Aedes aegypti IR7D does in fact respond to lactic acid stimuli in an in vivo model system. And then very, very broadly, Drosophila melanogaster can be used as kind of a tool and a vector for identifying all of these different insect taste receptors. So you can screen them in Drosophila before you even, take things to another model organism. And so some of my future directions, I obviously have to finish gathering that data to support the role for Aedes aegypti, IR7D, and IR56D in the behavioral attraction to lactic acid. Next, we want to know if these receptors can detect any other chemically similar substances. So lactic acid is a carboxylic acid, potentially, acetic acid, citric acid, some of these other acids, and then also fatty acids. These are found on the skin surface, and they could also be used as host cues. So it would be good to know if IR70 and IR56D detect these as well, or if they're specifically tuned to lactic acid. And then lastly, we're in the progress of generating a mutant in Aedes aegypti for IR70, so that we can test everything in mosquitoes as well. And so, yep, that's my talk. Thank you so much for listening. I believe Jinfeng is next and is going to tell you about some connectome stuff.
Do you just connect this one? Yeah. So, hi everyone, my name is Jing Fan, and today I'll be talking about the coding of gustatory information in the Drosophila, which is the fruit fly, in their brain. So, there's no presenter view of this one. Let me just double check. Yeah, choose. So both vertebrate and invertebrate brains contain so many neurons. So for a human brain, there's more than 80 billions of neurons. And for the common fruit fly, the Drosophila melanogaster, there are still more than 100,000 neurons in them. And they are wired together to form the so-called circuits. And these circuits that drive behaviors, for example, feeding and foraging. And our lab is not only interested in how those taste buds are detected by the sensory neurons, we're also interested in how the downstream circuits are connected to form robust circuits to drive all these different types of behaviors. So today we'll just be focusing on the coding principles of this early layer, which is a layer right after the sensory neurons. In terms of coding strategies, there are two models here. So the label line coding basically shows that each taste quality is encoded by one single cell, which means that different taste qualities are processed separately. In terms of the combinatorial versus population coding, you can see that each taste quality is encoded by multiple neurons, which means that an ensemble of neurons are required in order to decode the incoming taste stimulus. And taste experts express calcium sensors in this area called gustatory afferents, and especially the geniculate ganglia, part of the chora tympani nerve. And they were trying to characterize within this layer of neurons, what is the coding principle? One study in 2019 found that majority of these ganglion cells are single in tune, meaning they are only responding to one type of taste. However, a following study in the following year found that majority of them, well, not majority, but one half of them are multiply tuned. So showing that in this study, it's showing that it's more conform to this label line coding, whereas in this study, it's in a large degree, it agrees to a combinatorial and population coding. And none of these studies have been done in flies. So I was very fortunate to use the recently published fly connectome, and it is based on the 3D reconstruction of the EM, the electron microscopy brain volume. And this is our starting point of exploring the taste coding strategies based on the connectivity among different populations of neurons. So we started by identifying the taste sensory neurons in the connectome. And they can be identified by their morphology, projection morphology, and neurotransmitter expression. So we're able to separate the 4 classes of taste sensory neurons in the connectome. One is bitter, and the other one is sweet water. RNA-4E, as Mike talked about, it responds to glutamate, and PPK23, which is the high cell neurons that Mike just mentioned. And for today, for simplicity, these are the types of taste neuron connectivity we'll be talking about. So one is a direct connection between the taste sensory neurons and the cholinergic local neurons in the SEZ. So these neurons drive feed-forward excitation to the downstream neurons. Similarly, another connection will be between the taste neurons, sensory neurons to the GABAergic local neurons, which drive feedback inhibition to the downstream neurons. And lastly, another type will be the connection between the taste sensory neurons and the taste projection neurons. These projection neurons, they receive inputs from the peripheral sensory neurons and send those informations to the other areas of the brain that for further processing. So we first examined the connectivity in using the connectome of the connectivity between the taste sensory neurons and the taste projection neurons. And the metric we use is called cosine similarity, which is a metric that measures the similarity of two vectors in an inner product space. And is a very common measure to investigate how similarly are these neurons connected between the two sets of neurons. So for example, each red little squares indicate there is common downstream targets. So a red dot here within the bitter box shows that the bitter sensory neurons all project to very similar downstream targets. And you see like seldomly a red dot in this area where the sweet and water and where a neuron of sweet and water neuron and the bitter neuron project to a common downstream target. So what we can see from this graph is that in all these red dots are enclosed in the box, showing that they all different types of sensory neurons tend to project to segregated downstream targets. Therefore, we think that within this population, the taste projection neurons, it more conforms to this label line coding model. We also examined the two other types of connectivity. One is with the excitatory cholinergic local neurons. And similar idea, we find that these red little squares are enclosed in a box, showing that it's still quite separately processed in the downstream neurons. Whereas for the GABAergic local neurons, the inhibitory local neurons, you're seeing more of overlap between the two classes of sensory neurons, meaning that the bitter sensory neurons and the IR94E sensory neurons that respond to glutamate project to common downstream targets. So for a little conclusion here, we found that cholinergic local neurons conform more to a level line model, whereas the GABAergic local neurons are more toward the combinatorial model, especially when you see the common downstream targets for the bitter and IR94E neurons. So, but however, this is just based on the connectome of one single fly. In order to verify, we did calcium imaging. So we're expressing the calcium sensors in either acetylcholine neurons or GABAergic neurons. So this is the data for the acetylcholine neurons. And what we are doing is we apply tastins that only activate a specific one type of sensory neurons and see what are the profiles of responses in those neurons in that CZ. So what we found that in those cholinergic cell bodies, about 2/3 of them are singly tuned, meaning they only respond to one type of taste. And about one third of them are multiply tuned, responding to more than two types of tastes. We also compared our calcium imaging with our connectome data, which we inferred based on just the pure sensory inputs, and they largely agree with each other. Moreover, the third bar here, we're comparing all these two data with another measure, which is from the leaky integrate and fire brain model. That is the model that models the neural responses based on all the connections in the brain, and it's based on the connectome. It was developed by Xu et al. And you can tell that roughly they're all three different measures agree with each other. Now let's turn to the GABAergic neurons here. We did the same experiment and we found that the data still look quite similar to the cholinergic data. And about like 2/3 of them are singly tuned and 1/3 of them are multiply tuned. And the 3 different measures still quite in large sense agree with each other. So in conclusion, the layer behind our sensory neurons, it adopts this model is a combination of this label line coding where each taste is processed separately, which you see in the singly tuned neurons. And it's also a combinatorial model because there's still quite a bit of portion that responds to multiple tastes. So what's next? A student in our lab called Carlos will be presenting you a model, a computational brain model called the leaky integrate and fire model. That was one of the measures I used to verify my tuning studies. And so this model is built based on the connectome, and it models the spiking activity of the neurons. And this can be used to predict certain behaviors based on the motor neural outputs. And I'll turn to Carlos to give us introduction of this model.
I just want.
So thank you, Jufeng, for giving like a brief introduction to kind of what I'm doing at this lab. So a little bit myself, I'm Carlos, and I'm an undergraduate student here at the Gordon Lab. And yeah, so I've been working with the Drosophila computational brain model and performing analyses of different brain circuits. So I'll give you a little bit of an intro on the model, and then I'll talk a little bit more about the analyses that I've been doing. So a bit of a brief introduction. So the functional brain model was built from the Flywire connectome, like Jinfeng mentioned, and that was developed at Princeton using a synapse level resolution, like electron microscopy map of like around 130,000 neurons. So then an artificial neural network was built by Xu et al. Using the brain connectivity and the brain anatomy, and was used to predict motor neuron outputs from visual, mechanosensory, chemosensory, and visual inputs. So a little bit of a brief introduction on a neural network in general. So a neural network is, like a computer model inspired by the brain. So it's made by layers of interconnected neurons, nodes called neurons. So this is obviously very inspired by biology. Each neuron then takes in information, processes it through like simple mathematical operations, and then it passes the result down to the next layer. So I have like a kind of schematic with different nodes, and that's kind of visualizing the connections within the artificial neural network. And as you know, like it's fairly similar to how the brain actually looks. So if you look on the left side, this is taken from the connectome data. So we see here, like, I believe this is motor neuron 9, and we see all the different synaptic connections to that neuron and their appropriate neurotransmitters. So in this case, each neuron in the model, so these theoretical nodes on the right, corresponds to a real fly neuron. And it's labeled as inhibitory or excitatory based on the neurotransmitter type. So the model labels a neuron as inhibitory or excitatory depending on whether the majority of synaptic connections use GABA or acetylcholine as a neurotransmitter. So mimicking biology, each neuron then has a resting potential and only spikes when activated directly. So you can choose to activate specific subsets of neurons directly at whatever frequency you want. And is also can be activated when upstream neurons, as defined in the connectome, cause the downstream neuron to reach the firing threshold. So this is taken from Shu et al.'s paper. And this is kind of a schema showing the model's proposed neural pathway from the activation of sugar neurons, so periphery neurons, from a fly tasting sugar. So this would be, I guess, yeah, when Mike was talking about when the fly is like walking over food and just determining if the fly would like to eat it. And as we can see here, we have like the second order and the higher order circuits, which Jinfeng was doing analyses on, and how these all relate to activating motor neuron 9, which initiates feeding behavior, known as like proboscis extension. So this is a little bit about why we care. So as we know through our like daily interactions with food and drink, taste and smell are very closely linked. So olfaction plays a large role in taste. And we seek to investigate how the activation of different sweet receptors and different olfactory receptors can result in changes to feeding behavior. So if the fly is tasting something sweet, but smelling something with a maybe an adverse smell or a smell that might be corresponding to some food, like does that affect how the fly may feed? So this is a little bit of data that I've gathered from running the model itself. So a little bit to note is that when the model runs, it runs 30 trials, and then you'll be able to you'll see the output of the mean and the standard deviation. So when we take a closer look, we see the activation of motor neuron 9. That's what we're graphing here in hertz. And then as a control, we have that dashed line in the middle, which shows just the activation of sugar neurons at 45 hertz. And the shaded area corresponds to a standard deviation. So as you can see with all the solid lines, these are the activations of motor neuron 9 based on co-activation of sugar at 45 hertz and different olfactory neurons at the corresponding frequencies you see on the x-axis. So as we can see, there's a portion of olfactory receptors that inhibit, seemingly, MN9 activation. So in vivo, neurons often experience spontaneous firing. So what we noted in this model, and based on our results, that it seems like different ORN co-activations inhibit MN9 activation. We predict that the model does not account for spontaneous firings, and this results in our deviation from what is seen in vivo in certain neural pathways. Since the default neural firing rate is assumed to be 0, so even though it has a resting memory potential, due to computational limitations, the model doesn't predict like random firing. So this kind of results in failure for specific double inhibition pathways. So instead of what you would expect double inhibition to potentially result in an excitatory pathway behavior or excitatory pathway circuit, the model often incorrectly predicts inhibitory pathways. So that's what we suspect we saw with all the inhibiting of MN9. Oh, so yeah, that's a bit of what I was just talking about. So given this, we decided to kind of take in a different investigate some different circuits and see if the model can correctly lead to some further insight in different pathways. So my current investigation is working on how does neuron activation influence egg-laying behavior. So we've identified these IR94E receptors, and there's a few papers that show that they're necessary for egg-laying behavior in vivo. And I'm currently working on trying to replicate that in silico and also kind of gain more insight on the egg-laying pathway. And yeah, that's what I have.
Everybody, so if there's any questions for any of us, I think we have a few minutes.
So interesting stuff, like you have introduced the soil, you can also their responses. So how do you, because right now our diet is like into the behavior of our health.
Yeah, so in case people couldn't hear on Zoom, oh, yeah, in case people couldn't hear on Zoom, basically the question is, these days we eat a ton of salt and sugar, like the Western diet is full of this stuff because it gets us to eat lots of things that people are selling. And so how does this high consumption of salt like affect our own perception of salt? And yeah, it's an interesting question. And it's hard to compare directly because what we experience as humans is like really long-term chronic exposure to high levels of salt, right? And so how that affects the perception of salt compared to sort of these more short-term changes in salt intake that seem adaptive to like basically turn up and down an animal's response to salt based on how much it's recently eaten. It's maybe different things going on. So yeah, it's an interesting question. How would chronic like full life exposure to salt change things? And I don't really have a good answer for you. We continue to be quite attracted to it, obviously, which is why people keep putting it into food. And we do know that in humans, if you salt deprive people, salt does become even more attractive. But yeah, the effect of this long-term exposure, I don't think it's really super well known. Thanks. Yeah, Ethan.
Hey, Mike, that's really, really cool stuff. So I have a question about the injecting the salt in the abdomen
Yeah.
experiment. So how do you think that works? Do you think, is it there, because there's also like diuretic hormones. There's like a lot of other stuff that can float around. Do you think it's through like a hormonal kind of response, or do you think there's something else going on?
Yeah, so our working model has been that it's some sort of hormone-like response involving a neuropeptide. So there's some sensor for salt levels circulating in the hemolymph that sends a signal, probably a peptide, to the brain or to the peripheral sensory neurons that then changes their activity. That's what we've always sort of presumed, but we have yet to find that signal. Yeah, so we don't know for sure. Yeah. Yeah, go ahead.
Yeah, thank you so much for the great presentations. I'm curious about the receptors for the lactic acid ones. There's one from Drosophila and one from the mosquito, and they both sense lactic acid. I'm just curious if they're related, like phylogenetically closely related, if they use the same mechanism for the lactic acid detection, or if they've independently evolved ways to detect lactic acid.
Zach, you want to turn on your microphone and just press that button?
So
Yeah.
So the sad story with comparing the Drosophila and the mosquito receptors based on sequence and phylogeny of just the genetic sequence is that the ionotropic receptors it's just a extremely highly divergent family of receptors. And so there likely isn't an exact one-to-one ortholog based on sequence alone. So unfortunately, looking at just the sequence itself, we can't really assume phylogeny, and we can't really get any good hits out of comparing sequence from melanogaster to mosquitoes, which is why we went for the protein folding approach, because that's essentially the only way that we can find something that responds to lactic acid, hopefully. But yeah, I think that's essentially the way that we had to do it, just because of how divergent the family is, and there's not a direct one-to-one ortholog by sequence, just based on what we know so far.
Yeah, the co-receptors are really well conserved, but beyond that, it's a lot of differences, unfortunately. Yeah, in the back.
About the location of the taste, so if there are some taste sensors and some of them late, are they
Yeah, that's a really interesting question. So yeah, like how, if the fly is tasting on the leg versus the proboscis, what does that mean to the fly? Like, what's the difference between those two things? And I think you'd get a different answer depending on the very specifics of what we're talking about, whether it's sweet or sour or bitter or whatever. People have looked at this in some contexts. And the overall take home is that taste on the legs, appetitive taste like sugar, is thought to be involved in initiation of the extension of the proboscis to then initiate feeding, right? On the labellum can also do that. But then there's also neurons in the pharynx that detect taste as they're flowing through the early part of the digestive system, which are more sort of involved in sustaining ingestion. Now, the neurons on the legs are also involved in other things you might expect from stimulation of the legs, like the fly will stop, for example, if they taste sugar on their legs. So there's certain motor programs that are specific to the different areas. Yeah, so there is some specificity, and we know that the downstream neurons in each case are distinct. So there's certainly like the circuitry to